Started with day 3 of the AIRR community meeting. So before I introduce our first speaker, I had just a couple of announcements that I wanted to make. So as everyone knows, this is a very exciting area. That's why we're all here. There's a lot of emerging opportunities. And to that end, there's a couple of job announcements. So for all the sort of students and graduate students and postdocs out there, we got a couple of faculty positions that are open that I just wanted to mention. So first, there's a tenure track bioinformatics PI position that's available at the NIH Vaccine Research Center. And Danny Duick, if anybody is interested in discussing that, please see Danny. I also wanted to mention at the Yale School of Medicine, we also have a search going on now in the Department of Immunobiology for a computational immunology, again, a tenured or tenure track faculty position. So if anybody is interested in either of those positions, please see me or please see Danny. And with that, I will just introduce our first speaker, John O'Malley from the Brigham and Women's Hospital in Boston, as well as the Dana-Farber Cancer Institute. And he's going to be talking to us about sequencing in CTCL.
Can everyone hear me okay? Exactly. Always a good sign, right? Perfect. It's funny, I was just teaching the medical students, and someone would always do something like that whenever I did it. But since I'm vertically challenged, I may actually do most of this over here so all of you can see me. But my name is John O'Malley, and I am a dermatologist that works at both Brigham and Women's Hospital as well as Dana-Farber. And my clinical and research interest is cutaneous T-cell lymphoma. And when I first joined the Brigham 3 years ago, I don't have any disclosures. But when I first joined the Brigham 3 years ago, I started working in the laboratory of Dr. Rachel Clark and our chairman, Dr. Tom Kupper, are world experts in cutaneous T-cell lymphoma. So I felt very fortunate to be able to work with them. And over these 3 years, I've started to transition to my own lab. But the work that I'll be presenting is kind of a culmination of the 3 years of work that we've been doing. And we have a great team at Dana-Farber that consists of 4 dermatologists, a radiation oncologist, a medical oncologist, and a bevy of clinical research coordinators. And we've had an IRB approved observational study now where we have been collecting skin and blood of CTCL patients since early 2000. So we have amassed quite a lot of clinical tissue, and it's due to the generosity of these patients that I'm able to present the data that we have today. So as a way of an introduction, cutaneous T-cell lymphoma is a rare non-Hodgkin's lymphoma of skin tropic T-cells, of which mycosis fungoides and leukemic CTCL are the 2 most common variants. Leukemic CTCL patients present with diffuse erythroderma, or red all over. Their malignant T-cells can be found in the blood, skin, and lymph nodes, and these patients generally do poorly. When we studied the malignant T-cells from these patients, they are thought to be derived from skin-tropic central memory T-cells, characterized by surface expression of CLA and CCR4, which allows them access to the skin, and L-selectin and CCR7, which allows them to transit between the blood and lymph node. Mycosis fungoides patients, on the other hand, present with well-demarcated erythematous patches and plaques. The malignant T-cells in early stage patients can be found only in the lesional skin, and these patients generally do well, but 20% of these patients go on to have systemic, often fatal lymphoma. Studying these malignant T-cells, they are thought to be derived from resident memory non-recirculating T-cells, and they are characterized by surface expression of CLA and CCR4, as well as CD69, which results in these fixed skin lesions. And even when we assay the blood in these early stage patients, we cannot find the malignant T-cell clone. So I'm going to be talking a lot today about early stage patients and the clinical challenges that we run into caring for these patients. So I wanted to briefly go through how we even stage the patients. So we use a TNM classification. And generally when we stage the patients, we're actually looking at how much the rash is affecting their body surface area. Their stage gets worse once it goes from patches and plaques to tumors or erythroderma, or if they have nodal involvement or visceral involvement. And as the stage increases, the prognosis worsens. So the stage at initial diagnosis is what we counsel the patients when we sit down with them when they first come in with a diagnosis of CTCL about how they are going to do with this disease. But one of the biggest challenges that we have, as you can imagine, is these patients come in with rashes that mimic other rashes that we commonly see as dermatologists. So differentiating CTCL from benign inflammatory dermatoses is a significant problem. And to put this into context, on average, from when the rash starts in these patients, it takes over 6 years for them to finally get a final diagnosis of CTCL. So before we can even sit down and prognosticate with the patients about how they're going to do, often we'll have a high clinical suspicion that it's CTCL, but all of the factors are not coming into place to give them that final solid diagnosis of CTCL. And part of that are the factors have to fit together. So the clinical appearance, there has to be suggestive histopathology. And since this is a cancer of T cells, we often look for a clone in the skin or the blood. And the current clinical gold standard is using a TCR gamma PCR. And while this is a very good study, there's a false negative rate of 25%, a false positive rate of 15%, so it's not perfect. And some of the inherent challenges that we have with the V-gamma PCR, I'd like to illustrate here. So this is taking V-gamma and J-gamma primers, amplifying it up, and then separating out the products of that PCR amplification by size, by electrophoresis, and then using densitometry to see how dark the band is, essentially. And looks like we are stuck on a timer. We'll go with it. So when we do the PCR, this is an example of where we actually have a patient with blood disease as well as skin disease. And the sorted clone shows two very clear peaks. In the peripheral blood mononuclear cells, the Phi-gamma PCR also works very well and shows the same two clonal peaks. But this is what happens often in the skin. When we look in the skin, we know where the peaks are supposed to be, but there's also oligoclonal expanded T cells that make the diagnosis much harder to render. And we wondered whether newer technologies like high-throughput sequencing of the T cell receptor will help mitigate some of these challenges. And we entered into a collaboration with Adaptive Biotechnologies a few years back. And I know Dr. Robbins spoke yesterday, but we've also worked closely with Dr. Kirsch, as well as the rest of the Adaptive team, where we have sent them skin and blood tissue specimens, and they have performed immunoseq. And the rest of the talk today will be on data from their assay. And the way that the high throughput T cell receptor, I'm speaking to a group of experts about this, but I just want to mention that with the adaptive normalization, so essentially spiking in a synthetic immune repertoire where they know how much they actually put into the assay reaction, and they've done a very good quality control check to make sure that the primers are all amplifying at similar frequencies, we can actually start to quantify the T cells, the amount of T cells in the skin, as well as how much the malignant clone is taking up in the skin itself. And here's a typical example of a lesional biopsy from an early stage patient with a patch. We generally see a monoclonal expanded T cell, which is the malignant clone, and numerous benign infiltrating T cells. So when we embarked on this study, what we really wanted to do is to see whether high throughput sequencing could differentiate CTCL from these other benign inflammatory dermatoses. And the first way we really wanted to assay that is actually just taking the top T cell clone that we found in the skin and to see whether it differed between rashes like psoriasis and patients with CTCL. And within our CTCL subgrouping of the 46 patients, approximately a third were early stage patients, but all stages were represented. And briefly, to define how we determined the top T cell clone is based 3D histogram, the amount of the top T cell clone sequencing reads divided by the total reads were essentially how we first looked at it. And often when we look at our CTCL patients, we see a very high clonally expanded cell that drastically is enhanced compared to the next highest T cell clone, which looking at other benign inflammatory dermatoses, when we had looked, we did not see this huge expansion of cells. But when we looked at the top T cell clone as a percentage of total T cells, we were surprised to see that there was a lack of clear discrimination between CTCL patients and the other benign inflammatory dermatoses. And looking at the aggregate data, Although there is statistical significance between CTCL and these other diseases, there was a complete lack of discrimination. It is not a very good test. So we began to reconsider how to think of malignant clone burden in the skin of these patients. And there's really two ways of considering this burden. One is the clone as a percentage of total T cells, which is what you just saw. The second is a clone as a percentage of all nucleated skin cells, including keratinocytes and fibroblasts, and being able to assay the fraction of the clone that makes up all nucleated cells, we hypothesized may be a better way of capturing the malignant clonal burden. And by looking at the top T cell clone as a fraction of all skin cells, we saw a much better discrimination between CTCL and the other benign inflammatory dermatoses. In aggregate data, we see that CTCL can be much better enhanced. I acknowledge that there is this overlap zone, but the vast majority of the patients we could differentiate between CTCL and the benign inflammatory dermatoses. All right, so we felt comfortable with that finding, but what we really wanted to assess was how high throughput sequencing compared to the current gold standard of TCR gamma PCR. And briefly, I just want to say that we looked at the beta sequencing, so we also did the gamma sequencing, but since this is a malignancy of mature alpha beta T cells, the majority of those cells have two gamma rearranged alleles, which makes analysis possible, which we did, but the beta sequencing is much simpler because often it was just one sequence that was amplified. We looked at 39 patients that had classic CTCL skin lesions and subjected the DNA from those lesional skin biopsies to both high throughput sequencing as well as multiplex PCR. In all 39 patients, we were able to identify a clone by high throughput sequencing, but 10 patients were non-clonal by the multiplex PCR. And when we went back and looked at the characteristics of those early stage patients, we found that 9 out of 10 were early stage patients. So we originally hypothesized that perhaps the reason that these were negative for multiplex PCR is because these patients, just by virtue of the fact that they're early stage patients, had lower malignant clone burden than the other patients. But when we went back and actually looked at the fraction of nucleated cells between the PCR positive as well as the PCR negative patients, what we saw is a similar distribution of the malignant clone burden in both the PCR positive and PCR negative patients, suggesting that for reasons other than malignant clone burden, the PCR was negative. The TCR gamma PCR was negative. And here's a couple examples of PCR negative patients and then the high throughput sequencing that we got, which showed a clear clone. Here's another patient with classic skin lesions of CTCL, and then another high throughput sequencing where the clone was at a very high burden. So the clinical advantages that we have found so far in differentiating these patients who are early stage or before they've even gotten the confirmatory diagnosis of CTCL is that high throughput sequencing really does help us determine clonality and helps to finalize the diagnosis in the context of these other factors that I had discussed. That the discrimination was most effective when the percent of fraction nucleated cells was used, and it can facilitate the diagnosis of early stage disease, so who cares? The clinical implications, though, of being able to go from 6 years and any improvement that we can be, I think is significant. We now have FDA approved as well as non-FDA approved therapies that we know that are specific for CTCL that can actually induce longer term remissions. There's no curative therapy for CTCL other than stem cell transplant, but there are therapies that can induce long-term remissions. And so this would allow us to actually use nitrogen mustard and low-dose radiation, which work very well in these early stage patients earlier in their disease course, and then it will take a prospective analysis to see if we're impacting outcome. For the second half of the talk, I'd like to discuss a story that is our kind of newest story that we found in the lab of how we've predicted early stage patients who have progressive disease by using high throughput T cell receptor sequencing. I want to bring us back to this Kaplan-Meier curve of stage at diagnosis. It's a strong predictor of prognosis, and it's actually the best predictor of prognosis that we currently have. But what we often find in the clinic is within stage patients, so those 20% of patients I was mentioning at the very beginning that ultimately have progressive disease come from all stages, and a lot of those patients come from the stage 1B, and occasionally stage 1A patients have this progressive disease. Now, I'll give you a clinical example. Both patients presented to us with stage 1B disease with similar body surface area, similar laboratory studies. The patient on the left did extremely well with narrowband UVB, almost achieved a complete remission, and has not progressed in the 10 years that we followed the patient. Patient on the right, 2 months after those pictures were taken, developed tumors in the groin region. 6 months later, PET CT scan identified visceral metastases as well as lymph node involvement in the inguinal chain. And currently, there is absolutely no test that can predict which of these patients are going to act. Even they presented the same way. One of them progressed, one of them did not. So we became very interested in seeing if we could develop a biomarker or find a biomarker, better said, in predicting within stage how patients are going to do, and especially these early stage patients when it's easier to control their disease, be able to predict that. So with this observational trial that we had, we went back and we identified patients that had a lot of clinical data in their chart where we could clearly identify whether the patient progressed or whether they had stable disease. And we developed a training set of 208 CTCL patients with a median age of 62 years, more men than women. All stages were represented, but 2/3 of the patients were early stage disease, and the median follow-up time was 9 years. We originally thought this biomarker was actually going to be gene expression signatures because prior literature had shown that these patients, there were 78 genes that were really prognostic in how these patients did. And so we first wanted to identify whether we could use fixed formalin paraffin embedded tissues using nanostring to see if we could almost recapitulate that old study. And we also did TCR beta, CDR3, or high throughput sequencing, again, through adaptive biotechnologies. And the way the study was set up is we took the earliest lesional biopsy with histology confirming CTCL, because again, we would love to be able to have the patient when they first come in, be able to do a biopsy and be able to help risk stratify them. We had 57 progressors, 167 non-progressors. Doing the nanostring analysis, unsupervised analysis, the patients clustered into 3 spontaneous groups. And there was one group that had much worse survival with a median progression-free survival of 50 months compared to the other 2 clusters. And we were very interested to see what genes were differentially regulated in that cluster. And almost all of the genes were T-cell specific genes, including cell surface markers, members of the JAK-STAT pathway, and the TCR signaling pathway. So we initially just said, well, that probably is because there's more T cells in the patients in cluster 1 and then in the other 2 clusters. And while there is a trend of more T cells compared to the other 2 clusters, it was not statistically significant. We continued to look at the data, and what we actually also found was that the tumor clone frequency, so looking at the clone as a percentage of the total T cells, so the top productive TCR beta sequence divided by all of the sequencing reads, that was more significantly associated with the poor prognostic cluster than the other 2 clusters. And when we looked to see how many of the genes correlated with the malignant clone of the 78 that we looked at, 38 out of the 78 genes correlated with this progressive disease group. And so we hypothesized that the malignant clone frequency could actually be used as a prognostic marker for patients at high risk of disease progression. And when we were looking at the skin stage, so again, looking at 1A, 1B tumors and erythroderma, in these early stage patients, the malignant clone frequency, we were interested to see that there was patients that had high malignant clone burden, even in the stage 1A group, as well as the 1B group. And the tumors, as we would expect, actually had a higher average malignant clone burden than the other skin diseases. But in the early stage, at least, the malignant clone frequency was independent of the skin stage. So we wanted to compare known prognostic risk factors to this malignant clone frequency to see if we were barking up the right tree in terms of it actually being a prognostic indicator. So we looked at both progression-free survival, overall survival, and did a univariate analysis. And those that were significant, we then did a multivariate analysis with these. And so age was a risk factor that we looked at, stage, LDH, large cell transformation, which is essentially when you take a biopsy and there are very large pleomorphic looking cells underneath the microscope. That is a significant adverse risk factor. And malignant clone frequency. And so the malignant clone frequency was significant in both progression-free survival as well as overall survival. And looking at the Kaplan-Meier curve for this, the malignant clone frequency in the original multivariate analysis was a continuous variable. We essentially, for a biomarker, wanted to make it a dichotomized variable so that one would be better than the other. And when we did a log rank test, 25% clone burden seemed to be the best cutoff point. And when we looked at the Kaplan-Meier curves for this, there's a hazard ratio of 4 in the progression-free survival and 4.8 for the overall survival in this cohort. We then internally validated the model using bootstrapping and then took a new cohort of 101 CTCL patients and looked to see if we could actually verify our findings from the first model using the model that we had done. And again, it did verify that the patients who had malignant clone burden greater than 25% had a significantly worse progression-free survival than patients who had a clone less than 25%. Now again, we were looking at the vast majority, 2/3 of the patients were early stage, but there was later stage patients which could be influencing the results. And so we went back and we analyzed only early stage patients, 1A, 1B patients. And what we found, again, is that this held true. So a clone greater than 25% showed significantly worse progression-free survival, overall survival, and was validated again in the early stage mycosis fungoides patients. And this was looking at 141 patients in the discovery set and 69 patients in the validation set. So to compare apples to apples, in these early stage patients, we looked at progression-free survival with all the known factors, again, that were prognostically poor for these patients. And what we found essentially is that malignant clone frequency greater than 25% as a single test was the most robust marker that we had that was predictive of the patients at highest risk of disease. So let me put it in the context of some numbers. So patients who progress, the higher the malignant clone frequency was inversely correlated with the time to progression or death. And essentially, an early stage patient that had malignant clone frequency less than 25%, 91% of those patients were alive at 5 years. For a patient that had a malignant clone frequency greater than 25%, only 46% were alive at 5 years. So while not a perfect biomarker of disease progression, we do feel that this could be a reliable marker, especially if it is vindicated in an independent cohort outside of our group at Dana-Farber, and if we can prospectively also show this. But having a reliable marker can improve the risk stratification for patient prognosis, can improve clinical trial accrual if you are testing a drug that you would like to see really does impact outcome. It allows us to do earlier institution of more definitive treatment, potentially more aggressive treatment. And if we can even make this biomarker better, it could potentially give us a more timely identification for patients to receive stem cell transplants, during which at a time in their disease when we can more easily control, because the success of the transplant is absolutely hinged on the fact that they're in complete remission when they go into transplant. So to conclude today's talk, high-throughput sequencing provides a precise and quantitative measurement of the malignant T cell burden in CTCL skin lesions. And we looked at this in kind of two different ways. Clone as a percentage of all skin T cells facilitated the diagnosis of the disease. And clone as a percentage of all T cells was predictive of disease progression, or at least showed us the patients at highest risk for disease progression and at an early stage. So with that, I'd like to acknowledge my mentor, Dr. Rachel Clark, and our chairman, Dr. Tom Kupper, who is also my co-mentor, and then especially Adele, who was a postdoc in Dr. Kupper's lab, and we had split the work talking about the second half of the talk with the biomarker for the patients at risk of disease progression. The Dana-Farber Cutaneous Oncology team, Adaptive Biotechnologies, and our funding agencies, including the NIH. Thank you very much. I'd be happy to take any questions.
Great talk. You described improving the biomarker. Can you suggest what that would look like? Is there some sort of more detailed analysis that you would want to be able to do to come up with a more clear diagnosis?
Sure. Yeah, so there's a couple of things that we're actively investigating in the lab. And when I was making this talk, I was thinking to myself, I'm so centric compared to this group that it's like looking at the whole repertoire. So we have not forgotten about the whole repertoire. And so there's a lot of benign T cell analyses that we're actually currently doing. And then in conjunction, the sequencing doesn't differentiate CD4 versus CD8 positive T cells. And most of the patients, it's not always, but most of the patients, this is a malignancy of CD4 positive T cells. And we have some findings that the CD8 positive T cells, the amount and the type of CD8 positive T cells are extremely important in how those patients do over long term. So we're hoping to be able to do some additive measures to this.
Hello, thank you. I've got a question. If you take biopsies from different locations, does that look similar or different, or is that any better or worse?
No, so it's a great question, and it's an important question. So in general, us dermatologists, again, like people roll their eyes, like we're concerned about patches, plaques, and tumors. So the thicker the lesion. So in a patient, sometimes they have patches and as well as plaques. So when we see a patient, we know the money is in the thickest lesion. So we're generally taking a biopsy from the thickest lesion. But if we took a biopsy from a patch and we took a biopsy from a plaque, the malignant clone burden is different. The clone burden is higher in the plaques than in the patches. But if we take biopsies from a patch and a patch, we have found similar findings in both of those.
You mentioned the patient's experienced remission. Can you say anything about the T cell landscape during remission versus disease?
It's a great question. Another active area of investigation. Suffice it to say, it's interesting. We started to do treatment efficacy monitoring of these patients. So we'll take a biopsy before and after therapies. And we're in the beginning stages of this, but some of our therapies actually appear to be cidal to the T cells where they cannot be found on standard survey sequencing after treatment. While other therapies, the skin looks perfect, normal, clinically totally normal, but the malignant clone burden is actually very similar to the time that they've had. Now, you have to think about frequency. When we're dealing with frequency, we're dealing with, it could be a different total number of T cells, and that's what we're finding. There's less T cells, but all of the T cells kind of went down in the same ratio, as opposed to a malignant clone going down preferentially compared to the rest of the benign T cells. Thank you very much, guys. Oh, yeah, please.
One more quick question. So you mentioned actually, and sometimes you have been taking biopsies from multiple different patches. Is it the same T cell clone that spans those patches?
Correct. Yes. Yes. We would actually worry about our diagnosis if it was not. But yeah, and essentially we've worked with Adaptive. So now we actually do this as a clinical test. So when they come in, we send it, it's CLIA certified, and we are able to identify the CDR3 sequence and potentially follow it over time if they do end up progressing. But it is the same.
So in the patients with mycosis fungoides, the 20% or so that might progress, do you ever see mobilization of the resident memory cells into the blood?
So that's another area of investigation, right? So it looks like it's derived from a resident memory T cell, but we know eventually it becomes systemic. So what's going on? Like, why are they changing? Occasionally we find 1 or 2 copies of the T cells in the blood. And again, this is standard survey sequencing. So we're not doing the deep sequencing that Dr. Crow was doing, but we can occasionally find it. So we hypothesize maybe that could be an indicator of disease progression as well. Thank you.
No further questions? All right, it's my pleasure to introduce our next speaker, Johanna Olveus, who is from the Oslo University Hospital and the University of Oslo, and she's going to talk to us about neoantigen-specific T cell receptor repertoires.
So first, thanks to Jamie and Felix for inviting me to this exciting meeting. I really enjoyed all the excellent previous talks. So I will talk about how T cell receptors can be applied in cancer immunotherapy and the potential advantages that donor-derived T cell receptors might provide in this regard. Okay. These are my disclosures. So this is the Institute for Cancer Research in Oslo at night, and here it is during the day. In the background here, you can see the old clinical building at Campus Radium Hospital, which is entirely focused on cancer. And here is the newest addition to the campus, which is Oslo Cancer Cluster and Innovation Park, that is dedicated to commercialization of cancer research and the home of some 50 companies. And it's also the home of a senior high school where we train the next generation of scientist broilers. So where are we today in terms of identifying good targets for cancer immunotherapy? So some people would argue that this represents a good illustration of where we are. This is the classic painting by Norwegian painter Edvard Munch, and he entitled the painting The Scream of Nature. So cancer, but there are still a number of challenges. And one of the reasons is that checkpoint inhibition relies entirely on the endogenous T cell repertoire for efficacy. And we still don't know what the T cells see in when we treat with checkpoint inhibitors, which makes the effect unpredictable. Furthermore, not all cancer types do respond, and a large fraction of responding patients do eventually relapse. So to know why that is, we need to know more about what the T cells can see in cancer. And it's known now that the mutational burden varies greatly in different cancer types, and that the cancer types that do respond the best to checkpoint inhibition have the highest mutational burden. It's also known that in the cancer types that do respond well to checkpoint inhibition, there is a correlation between the mutational load and durable clinical benefit, as shown for melanoma, for cancers with mismatched repair deficiencies, and as shown here for non-small cell lung cancer. So this is all indirect evidence that T cells can recognize mutations in cancer. By now, it has also been directly demonstrated by the Rosenberg group that T cells can have tumor mutations as rejection antigens. So expanded T cells reactive to a mutant peptide derived from the ERBB2 interacting protein mediated regression of cholangiocarcinoma that was metastatic in one patient, and in another patient, expanded T cells recognizing a mutation in KRAS mediated regression of metastatic colorectal cancer. But the question is, are these exceptions, or do they represent the rule? So we looked into the literature to see how frequently do T cells spontaneously recognize mutations in cancer. And the studies that are listed here did either an unbiased screening of T cells that could recognize mutations in cancer, or they selected peptides that were predicted to bind to the HLA of the patient and screened those. And to summarize, the hit frequency was very similar in all of these studies, and T cells responded spontaneously to 68 out of 5,842 mutations that were screened for in 36 patients included in 11 studies, which gives a hit frequency of 1.2%. So 3 other studies are questioning the ability of the patient immune system to permanently erase cancer, and that includes 2 recent reports in Science in 2016 and one in Nature in 2017. So these studies argue that there is an epigenetic reprogramming going on in tumor-reactive exhausted T cells that makes them permanently dysfunctional, and that treatment with anti-PD-1 only temporarily can reverse the functional state. So we asked the question, can the immunogenicity of candidate neoantigens be evaluated independently of the immune response of the patient? And can T cell responses to neoantigens be broadened independently of patient immunity? And to this end, we interrogated the T cell repertoires of healthy donors because they are independent of coevolution with cancer. And this project was performed in a very nice collaboration with a group of Ton Schumacher at the Netherlands Cancer Institute. And the 2 guys that did the large part of the hard lab work were Alan Strenen, MD PhD student in my group, and Mireille Toubs, engineer in Ton's group. So we started out by mapping the mutations that could be found in 3 HLA-A2 positive melanoma patients, following the general pipeline of exome and RNA sequencing, and after this predicted which of the identified mutations that were predicted to generate peptide that could bind to HLA-A2. And out of more than 300 candidate peptides, only 2 reactivities were found in the patient tumor infiltrating lymphocytes. So very similar to the data that I just showed you previously. So we next tried to set up a platform for culture of T cells from healthy donors to induce neoantigen reactivities. And we started out by generating constructs and then produce mRNA encoding the mutations as a string of beads. Then we introduced the mRNA into monocyte-derived dendritic cells by electroporation and co-cultured them with autologous T cells. And at the end of this co-culture, we detected neoantigen reactive T cells by use of fluorescent peptide MHC multimers. And the positive cells were sorted as single cells for cloning. And this shows a typical staining of using multimers of T cells that had been stimulated with the neoepitope mRNA. And the specificity of the staining is shown here as control culture that was stimulated with irrelevant mRNA did not stain for these multimers. And to the right here, you see the summarized results for healthy donor T cell reactivities from 4 different donors to neoantigens identified from patient 1. And what this shows is that the majority of the immunogenic neoantigens evoked a response in all 4 donors, and with all neoantigens evoking a response in 2 or more donors. And importantly, 4 out of 5 of these neoantigens were actually neglected by the patient T cells. So importantly, T cell clones that were generated recognizing these neoantigens from healthy donors responded to cognate patient tumor harboring the relevant mutations, whereas a third-party tumor that had the corresponding wild-type version of these genes were not recognized. And this shows a summary of some of the 300 clones that we tested, and it shows basically the same thing. We then sequenced 28 of the T cell receptors from the clones and tested their reactivity upon expression in peripheral blood T cells. This shows one example of a TCR reactive to a mutation in the MLL2 gene, and these redirected T cells recognize cognate patient-derived tumor with a mutant gene, but not third-party tumor, unless the mutant gene was introduced in the third-party tumor, and cognate tumor where the mutant gene was knocked out led to a much reduced response. And this graph here just shows similar results for another TCR responding to another mutation. So in conclusion, we think that T cells from healthy donors, that we've demonstrated that they can respond to a repertoire of neoantigens that are neglected by the patient T cells in vivo, and that T cells redirected with neoantigen-specific TCRs identified from donor-derived T cells efficiently can recognize patient tumor. And additional advantages are that T cells responses in this way are made independent of tissue and blood sampling from patients. And also that the neoantigen reactivity can be broadened by the use of multiple donors, since as I showed you, not all donors respond to all the same neoantigens. And with the only requirement that these donors are matched at least for one HLA allele with a patient. So we think this provides a rationale for the outsourcing of cancer immunity to blood donors. So an important next step to clinical translation is to find a good way of doing a high throughput identification of the best TCRs for therapy. And Wei-Wen Yang in my group has performed single cell TCR sequencing using an approach developed in Ton Schumacher's group. And this example shows that TCRs derived from different donors actually can have a high degree of sequence homology, as indicated by the colored regions here. And here is a simplified scheme of how we envision that healthy donor T cells can be utilized in a clinical setting or in a therapeutic setting. I apologize that the background is a bit pale here. But after identification of the expressed mutations, the mutations can then be screened for reactivity, or for the T cells of healthy blood donors can be screened for reactivity to these mutations. And the results can be used to identify either T cell receptors for use in adoptive T cell therapy, much the same way as CARs are used, or for identification of immunogenic neoantigens for vaccination purposes. And an indication that this might be of clinical value is the fact that there are now several companies that have taken up this approach, such as Medigene and Neon. So we also asked if we could improve the prediction of neoantigen immunogenicity. The hit rate that we got, we selected 57 epitopes that we tested for donor T cell reactivity, and we found responses to 11, giving us a hit rate of 19%. We then looked at the predicted binding affinity of these peptides, and we found that the peptides that did induce a response had a higher predicted binding than the ones that did not induce a response, which gives a lower nanomolar affinity value. But the difference was not significant. So based on a previous literature from microbial peptides, we asked whether measurements of peptide MHC stability could improve on the identification of immunogenic peptides. So we then developed a flow cytometry-based assay for measurements of peptide MHC stability that was bead-based. And using this, we could indeed show that the responder peptides had a significantly longer off rate than the non-responder peptides. And if you use this to set a cutoff that included all of the immunogenic peptides in red, we could actually exclude about half of the non-responder peptides, giving us a hit rate of 36%. This can be expressed in a different way using receiver operating characteristic curves, and this shows that excuse me, by addition of measured peptide MHC off-rate values, we can increase the area under the curve relative to use of predicted binding affinity only. So next, we wanted to investigate if this could be used to also improve on the identification of immunogenic neoantigens for peptides binding to other HLA alleles, where the prediction algorithms are actually less good than for HLA-A2. And we also wanted to test if we could improve the prediction of or identification of immunogenic peptides in a larger set of peptides that had already been screened for ability to induce immune responses in patients. So then we started out with a large set of peptides that were encoded by mutations that were identified in 9 melanoma patients from Ton Schumacher's group. And after identification of expressed mutations, peptides were predicted by Marit van Buren, and they were tested for ability to be recognized by tumor-infiltrating lymphocytes by Pia Quistborg. And we ended up selecting a sample of 487 peptides that cover the full range of predicted binding affinities, and that were predicted to bind to 5 different HLA alleles, and that included all the hits that were found in the patient T cells. And we used then this set for measurements of off rates with our assay. And the results are shown here. So the peptides that did induce an immune response in the patients are in red, whereas the non-hits are in black. And on the y-axis are the measured peptide MHC off-rate values, shown as ranked values, meaning that the peptides with the longest off-rates are at the bottom. On the x-axis are the predicted binding affinities using NetMHC 4.0, and here then the top predicted binders are to the left. So if we now use this to set a cutoff based on predicted binding affinities, we can exclude a large portion of the non-binders, but if we also include the measured peptide MHC stability values, we can exclude a much larger proportion of the non-binder, of the non-responder peptides. And this can also be illustrated again with a receiver operating characteristic curve, showing that addition of measured peptide MHC values increases the area under the curve. So we think that this showed that peptide HLA stability measurements can improve prediction of which neoantigens are immunogenic in patients and for multiple HLA alleles. So in the first part of my talk, I have discussed how identification of T-cell receptors from healthy donors that recognize neoantigens potentially can be used in immunotherapy. The challenge with this is, of course, that most mutations are private, meaning that any therapy involving such TCRs will be highly personal. An alternative strategy would be to try to identify shared mutations and then generate T-cell receptors that recognize these. And such a therapy could potentially be used in more patients. The challenge is that in solid cancer, there are very few recurrent mutations. So for instance, P53 is very often mutated in cancer patients, but the mutations are usually in different loci. In leukemia, there are far fewer mutations, but quite a few of them are actually shared or recurrent. So Irini Giannakopoulou in my group, she set out to generate a T-cell receptor to a shared mutations in acute myeloid leukemia. And by stimulating healthy donor T cells with dendritic cells that were then transfected with mRNA encoding the mutation, she generated a neoantigen positive population here. And the specificity is shown by lack of staining in a control culture that was stimulated with the melanoma neoantigens that I showed you in the previous study, and vice versa. And we then identified the TCR and used it to redirect peripheral blood T cells. And what this shows is that the T cell receptor is highly sensitive to peptide down to the picomolar levels when loaded onto 3 different cell lines. Here we show that the TCR is highly peptide and HLA specific, as a large number of cell lines that are of various tissue origins but that are all HLA-A2 positive are not recognized by the TCR unless the cells are loaded with the mutant peptide in red here, whereas there's no recognition when they're loaded with the wild type corresponding peptide in blue. And this shows that the TCR also recognizes endogenously processed antigen, as these cell lines here that do not express the antigen are transfected with mRNA encoding the mutation here. And then finally, we also showed that patient AML cells harboring the relevant mutation were strongly recognized, whereas 2 patients that had other mutations in the same gene were not recognized unless they were loaded with the relevant mutant peptide. So we think this might warrant further development towards the clinic. So in the final part of my talk, I will also discuss TCRs reactive to shared antigens. And the obvious advantage of targeting shared antigens is, of course, that then all cancer patients with a certain cancer type could be treated with such TCRs. The challenge is that the shared antigen will also be expressed on the non-malignant counterpart of the cancer cell, and therefore you could have severe on-target off-tumor toxicities. So an example where this is very well tolerated is rituximab or anti-CD20 antibodies, which are standard in lymphoma treatment, recognizing the B cell specific CD20 molecule. And the reason why it's well tolerated is that we can do without B cells. Another example is the CD19 CAR, providing means of making therapeutic antibodies more efficacious. So chimeric antigen receptors have, as you know, the extracellular portion of an antibody coupled to T-cell receptor signaling intracellular part. And CAR-19 therapy was FDA approved this summer due to the great success it had in treatment of B-cell malignancies. And there has also been an extensive search for CARs to other cancer types over many years with limited success so far. So this has also made us ask whether we could translate the success of CAR-19 therapy to T cells in terms of that we could identify T cell receptors recognizing self-antigens. And the advantage of T cell receptors in this regard is that they can also recognize intracellular targets and that would vastly increase the number of potential therapeutic targets. So the autologous T-cell repertoire is normally devoid of T-cell receptors that can recognize self-antigens with high affinity due to the process of negative selection. And this means that the autologous T-cell repertoire has limited utility for identification of T-cells and T-cell receptors that can recognize self-antigens with high affinity. In contrast, the T-cell receptor of an HLA mismatched donor has not been negatively selected on the same HLA, and therefore a T-cell can recognize a self-antigen presented on a foreign HLA molecule with high affinity. And the person who first suggested that this could be used therapeutically was Hans Strauss. So we investigated the breadth of the self-immunopeptidome and utility for T-cell targeting using HLA-A2 negative T-cells for detection of self-antigens presented on HLA-A2. And the culture platform that we set up is very similar to the platform that I showed you in the first part of my talk for neoantigens. We started out by predicting identifying cell type specific targets by searches in databases such as IST or GeneVestigator. And then we predicted which of the peptides from the candidate target proteins that could bind to HLA-A2. And secondly, we then generated panels of color-coded peptide MHC multimers that were complex with the different peptides. And to circumvent the fact that tolerance to self is restricted to self-HLA, we used T cells from HLA-A2 negative donors for detection of self-antigens presented on HLA-A2. And we did that by taking autologous dendritic cells that were then A2 negative and transfecting them with mRNA encoding A2 and also the target protein. And at the end of co-culture, we detected antigen-specific T cells by use of the color-coded multimers. And we would get dot plots looking like this. And we actually concluded that we could discover T-cell reactivity to 36 out of 50 predicted epitopes, which represents a really high fraction using non-tolerized T cells. And these epitopes were derived from 2 leukemia-associated proteins. We've published this. I'll just show you one example at the very end of a T-cell receptor that we have generated in this way that is recognizing a self-antigen in the context of allogeneic HLA-A2. And this T-cell receptor recognizes a peptide from CD20. And we've shown that it is highly peptide and MHC specific, and that it can also mediate killing of patient lymphoma and leukemia cells in these publications here. So why would a CD20 specific TCR be interesting now that we have CD19 CARs? Well, patients relapsing from CD19 CAR therapy are frequently CD19 negative. That has now been shown in many publications. And there was also now a recent publication in Nature Medicine where patients treated with CD22 CARs relapsed quickly. And it was shown that they were CD22 negative, but had unaltered mRNA levels for CD22. So the intracellular levels of antigen was there. It's also been shown that TCRs recognize antigen antigen levels that are much lower as compared with CARs. And of course, they can recognize antigen independently of cellular localization. So this means that TCR might provide an attractive alternative or to be used in synergy with a CAR because they can detect lower antigen levels. So this shows these TCRs in a mouse model. And this is work that has been done in collaboration with Matthias Leisegang at Charité in Berlin. So he has a great mouse model that is HLA-A2 transgenic. These mice lack murine class 1 molecules. It's a syngeneic immunocompetent mouse model. And what Matthias did was that he co-transduced a fibrosarcoma cell line, MC703, with constructs that encoded the CD20 epitope and the chimeric molecule HHD, which is a chimer of human HLA-A2 and the murine constant domains to allow binding of CD8 from murine T cells. And after having established tumors that were quite large, around 300 cubic millimeters, after 1 month, he injected murine T cells that were then transduced with the 2 different CD20 specific TCRs. And what these graphs here show is that the mice rejected the tumors, and at the maximum observation day, we actually have the remaining portion here also. At the maximum observation time of 100 days, the mice were still tumor-free, and they had no autoimmunity, which makes us quite optimistic, since the mice are A2 transgenic, and human and mouse are sharing quite a large portion of the proteome. So we think this is promising both for safety and efficacy. So the conclusions of this last part is then that the repertoire of self-peptides presented on HLA class 1 has been underestimated, and that alloreactive T cell repertoires can be utilized to efficiently and specifically target tumor-associated antigens. That CTL populations reactive to self-antigens can respond avidly and specifically to target cells expressing the corresponding antigens, including leukemia cells, and that this platform should avoid the identification of false positive epitopes and tolerance to self-antigens, and that T cell receptors reactive to a peptide from CD20 can eradicate large established tumors in murine A2 transgenic syngeneic tumor models. And we think the technology should boost the development of efficacious immunotherapy in cancer by identification of high affinity TCRs. This is my hardworking group. I'm indebted to them. And this is the acknowledgement slides. I would in particular like to mention the great collaboration we've had with Ton Schumacher and his group at the Netherlands Cancer Institute, with Fridtjof Lund Johansen and his group at Oslo University Hospital, and with Matthias Leisegang at Charité in Berlin. Thank you so much.
Yeah, so great talk, fantastic work, and I really mean it, because now I'm going to bitch about the part I hate. Can you go back to the conclusion slide where you compare stability predictions and binding predictions?
For the stability, okay. All right.
That one, 27.
20, let me see. Is it not the next steps, this one, the first, this one?
No, 27, slide 27.
Slide 27, okay. Slide 27 is not the conclusion slide. Okay, but this one?
Yes, okay. So the way you presented it, you said stability prediction outperform binding predictions to identify these.
Well, we use it in addition, right?
Okay,
so you
that's
kind
the
of
key
say,
point,
yeah.
because what you're showing here is you essentially have a threshold of rank about 2%. So in the top 2%, you have all epitopes, and in the top 20% in terms of stability, you have your other one. So, and I'm just.
That's why I also pointed out that it can provide additional value when you also start out with predicted peptides. Yeah,
that's also what we see whenever we do it. If you had started off with stability as a selection criteria for your peptides, because these are clearly pre-selected based on
absolutely
affinity, it would be exactly the other way around. And I'm just pointing it out because this is like a myth being propagated at this point by other people who are not as smart as you, who are just then concluding we shouldn't be doing any more affinity measurements or predictions. Stability is a nice addition to it, but it's the consensus that matters, not the.
Yeah, no, the combination is certainly the way to go, I think, too.
When you presented the pipelines, there was a word mass spectrometry appearing in the pipeline, but I wasn't clear how mass spectrometry is participating in this and what is the target database to search mass spectra. Can you elaborate? What is the role of mass?
You probably mean the summary figure that I had. Yeah, sure.
Overall slide.
No, that was up here. So that was just that as an alternative to just doing exome and RNA sequencing, you can also do mass spec to identify what is actually presented. But that doesn't mean that it's immunogenic, right? So even if you identify all the presented peptides, you need to have a way of identifying which of them are actually immunogenic. So the input for the test with a healthy blood donor T cells could be either peptides identified by mass spec or peptides identified by exome and RNA sequencing. That was just my point.
Are you doing mass spectrometry, or this is just?
No, we're planning to do it.
Can I ask, what was the age of the healthy donors, the age range? And number two, you think about this a lot. What do you think about loss of HLA on solid tumor cells as an issue in this type of set of goals?
Right. So the age of the blood donors, it's regular blood donors, which have, I think the average age is around, 30, 40 in the blood bank. So these are just Buffy codes that we get from the blood bank. So it would be quite a wide spectrum.
Not particularly on the young side.
No. And the other question was, sorry.
Loss of MHC on solid.
Also, yeah. Well, that is of course a challenge, and so that might be also that you have to combinations of TCR and CARs could be worthwhile just for that reason.
All right, I just want to make some announcements about scheduling. As some of you may have noticed, we inadvertently scheduled an hour and 25 minutes for Anaki's talk. And so what we're going to do is we're going to give a bit of a longer break now. So instead of having essentially a 15-minute break, we'll break now for I guess more than half an hour. We'll start up the next section at 10:30. So at 10:30, we'll have Anaki's talk. And then the talk after will be immediately following, so at 11:05. And then that will give us also some more time for the poster session and lunch. We'll have a lot more time to browse the posters. So 10:30, back here.
Sure, in the pocket, okay.
Okay, great. Well, welcome back, and have a couple administrative things. Here's an important one, will wake you up. They're going to remove the posters at 5 p.m. So during that last coffee break, around 3:15, make sure you bring down your poster or pick up your poster or whatever, because the poster boards, I guess, with posters will be removed at 5 p.m. So the dinner is on for tonight. We're hoping it won't rain. There's a map in here that it's one metro stop down to White Flint and a short walk. If it's raining, I warned the restaurant that there'll be a lot of wet people showing up around 6:30, but it's show up at 6:30, have dinner at 7. It's in the booklet. It's Seasons 52. And people have been talking about getting together. We have a big break at lunch, and having an informal group here at 1:30 to discuss the ideas behind the IP discussion that we had starting yesterday about recommendation 4 and everything. So be an informal group. So that would be, yeah. So, anybody who wants to could show up and discuss it more. Their system thinks it's gone forward a little bit more. So that would be 1:30 in here, an informal group, and then we'd start the agenda at 2 again in the afternoon. Thanks.
All right, so it's my pleasure to introduce our third speaker in this session, Anaka Sands from the Emory University School of Medicine.
Okay, thank you, Steve, and thank you, Jamie and Felix, for the invitation and a great meeting that you have organized. So as you can see in the title, what I want to discuss with you today is how we use different tools to understand human B-cell differentiation, pathways of activation, and how that may be abnormally regulated in human autoimmune diseases, and in particular in SLE. So this is very timely what Jim Crow was presenting last night, because these were some of the questions that he is addressing, I think, are very, very important, because they should provide the background that we need to then understand disease abnormalities. We really need to understand the normative rules for repertoire, as you see here under the questions. What is the complexity? What is the diversity, both in a global context or in antigen-specific settings? What degree of persistence, diversification, replacement of particular components of the repertoire may be taking place during active immune responses, and in particular in chronic autoimmune diseases that are also subject to flares and relapses. With the population connectivity, can we understand that through repertoire sharing of different clones? What is the directionality between different compartments, whether it's the cellular origin and diversification or compartment relationships in autoimmunity? Obviously, that's critical. What is systemic? What is in the tissue? What is being locally either originated or mature versus what is just simply passive migration from the systemic immune system. So on and so forth. And I think that in order to understand that, we need a lot of the information that was discussed yesterday. We need to understand and overcome all these challenges: cell numbers, depth of sequencing, accuracy, and quantitative aspects of the sequencing, as well as the biological issues. How do we really apply the repertoire analysis to the proper B-cell compartments with proper definition of those subsets? And then do we really have the right quantitative and qualitative samples that we need? Again, cell numbers, blood, or tissues. Timing will be essential, not only because we don't know the consistency, again, as Jem was saying, from day to day, but also longitudinally, obviously, as the disease changes. And unless we know what the normal variability is, then we are not going to be able to put that in context. Individual variability, and then obviously many other variables that we need to understand. So the way we approach understanding human B cells in disease is sort of summarized here. It's sort of a comprehensive approach that we have termed, not very originally, but B-cell immunomics for human disease. And this is something that we do internally, but in collaboration with Giorgio. This part of the figure actually is his own figure that I stole from a grant that we wrote together, and then integrated with the rest of the approach that we take. And then great collaborators internally, and now at UAB, Alex Rosenberg and Chris Ficilli, and then Chris Tipton in our group that does all the B-cell repertoire, Deepak Tomar, Chang Wen Wei for flow cytometry, and Jerry Bose for the epigenetic analysis. So essentially, what we do is to get the right tissues, starting with the blood, but also related tissues in SLE. For lupus nephritis, for instance, we take the serum odd antibodies, and again, this is George's approach to immunoproteomics after purification of the autoantibodies of interest, in this case for lupus. And then the immunoproteomics, we do then cell B-cell repertoire analysis by RNA sequencing, and then we do from the same B-cell flow cytometry to do a very careful definition of B-cell subsets I'll discuss in a minute, and then try to generate profiles of different subsets of disease. And then from those B cells of interest that we identify by flow cytometry, once we see perturbations that suggest that a particular B cell subset is important in disease, we purify those B cells not only for repertoire sequencing, but also for transcriptional and epigenetic analysis that I'll discuss at the end of the talk. We have used this approach quite successfully, both to define the diversity and origin of antibody-secreting cells in flaring SLE, as well as to define the origin of long-lived plasma cells responsible for antiviral responses after more than 40 years of exposure. And so this integrated approach has proven to be actually quite useful for our purposes. We have also published how the epigenetic analysis of naive B cells in lupus give us some insight into what's abnormal with those B cells, and I'll show you some of that. And then finally, now we are, as many people, moving in the direction of high-throughput single-cell analysis. We started doing that through George as well with his high-throughput emulsion PCR, which has been working quite well in the lab. And now we are moving in the direction of doing 10x analysis, both of the RNA transcriptome as well as the B cell receptor repertoire. And Steve Bossinger, who is here and has a poster later today, has developed a pipeline to actually reconstruct the antibody sequences from the single cell mRNA. This a collaboration that we have with George, which describes a little bit, illustrates what I just said. This analysis of lupus flares in patients with lupus nephritis, and as you can see, what we are trying to do is to assess the contribution of different types of plasma cells, either short-lived or long-lived plasma cells, to the lupus autoantibody proteome that is expressed in the serum, because in the end, that's probably what is more relevant to disease. And the design, as you can see here, is to take patients that have a flare of lupus nephritis or acute presentation, and for whom we have baseline, so prior to nephritis serum and cells, in some cases also from the bone marrow. We can obtain the same samples at the time of acute nephritis and then 1 to 3 years after flare, so that the integration of all this data presumably will tell us where are the new autoantibodies that are surging at the type of nephritis, whether or not they were present before, and whether or not they are sustained several years after the disease flare. So one of the issues that we need to tackle is the complexity of human B cells, which is an ever-growing issue. This is just a representation of the more conventional analysis by 3 or 4 or 5 colors of CD19 positive B cells, defined by CD27 and IgD, and you see the major parental populations that I think are well defined: the naive population and switch memory, the switch memory and the plasmablast, and then this double negative population that lacks CD27, so a marker of memory, and IgD, a marker of naive cells. They both are absent. And then you can do other type of gating to better identify the antibody-secreting cells. Now, if you do more colors and if you do multidimensional analysis, then you start recognizing, as you can see here, many more populations in the human blood and many alterations in lupus relative to healthy controls. And so we have reported on that for a while now and used those approaches to identify new populations in SLE, in particular these double negative populations that I'm showing you here, which are highly expanded in active patients, and I'll show you that as part of the talk. This is the work along those lines that we are doing with the WHO for the B cell nomenclature subcommittee, which is summarized in here, the components of the committee. And this is, I'm not going to go in any detail through this, but this is just an idea of the proposed nomenclature and the many subsets that I think can be consistently identified in the human blood and that represent, I believe, biological subsets of significance. And you could see that here, for instance, with the switch memory cells, where we can identify resting or activated naive cells, memory cells, the so-called atypical memory, the double negatives, which share some aspects of the ABCs or H-associated B cells, and then early activated memory cells, also called APCs for a different reason, that actually Scott Boyd and Rafi Ahmed described recently. So this just to give you a sense of where we are going with these, but it's my belief that we really need to be precise on how we identify B cells, and then consistent, ideally, from group to group, so that we can really understand the problem better. So in particular, let me tell you about these double negative cells and how we are trying to understand them. So this is just a cartoon showing, obviously, the two major pathways of B cell differentiation, starting with naive cells, getting activated, getting some help from extrafollicular T helper cells, and developing into activated naive cells and short-lived plasmablasts. This may lead to the germinal center reaction under the right stimulatory conditions, and then produce memory cells as well as long-lived plasma cells. So Chris Tipton in the lab a couple of years ago provided, I think, the probably better, or at least a good description of what the activated naive cells look like in lupus and how large the expansions are during acute flares. So here we are defining them with a particular set of markers. I'll show you the ones that I think that are better these days. And so we show that actually those activated naive cells were not only expanded in acute lupus, but also were highly connected by repertoire analysis through sequencing of the blood with the antibody secreting cell compartments also in the blood, either CD38 negative, 138 negative plasmablasts, or CD138 positive plasma cells in the blood. So then we, as I said, are interested in these double negative cells, which in lupus become extremely prominent. As you can see here in many patients, at least the acute flares may be the dominant population in the blood. You can see that these are patients also with massive expansions of plasmablasts. And so these double negative cells have been described with similar phenotype, at least, as early activated or even exhausted memory cells using different combinations of markers. And this is in particular the case for chronic HIV infection. However, we wanted to test the idea that, in fact, they might be in this pathway. And I'll show you evidence, hopefully, to convince you that that is the case. So when you start looking at SLE with different markers, I'm showing you here CXCR5 because I think it's highly informative. Again, you can see a major expansion of these double negative cells relative to healthy controls. And when you look at them by CXCR5 CD19, you see that there is a major fraction both of the double negatives as well as the naive cells that share this phenotype, which is CD19 high as a marker of activation and CXCR5 negative. Whereas in normal subjects, the vast majority of systemic B cells are CXCR5 positive. So this becomes a very helpful marker that allows us to differentiate now what we call DN1s and DN2s that are CXCR5 negative, CD19 positive or high. And here you have the extended phenotype of those cells, which I'm happy to discuss if anybody's interested later. But just to say that actually, with the exception of IgD, by definition, the activated naive cells and the DN2s share essentially the same surface phenotype, even this extended one. And I'll show you molecular evidence that, in fact, they are connected from a differentiation standpoint. Just also to make the point, because there are more and more reports every day of TBET positive B cells that people, I think, unfortunately, are lumping together under the label of ABCs, as if it was really a homogeneous population, when in fact, I think it's very heterogeneous. In our hands, at least, these TBET positive or TBET high B cells really concentrate almost exclusively in the activated naive cells and the DN2s, which also have high levels of expression of CD11C. This is the molecular analysis by RNA sequencing that we did of the different populations I really don't have time to go over these markers, which are very interesting, but I summarize them for you in a cartoon later. What I want to do is to make the point here over this data that the DN1s and the switch memory cells are almost identical transcriptionally. You can see that in here there is only about 20 genes different between the two populations. You see the many other differences between the other populations. When we actually sort the DN2s and the activated naive cells and we do the same type of analysis, now you see that the DN2s are also very close transcriptionally to the activated naive cells, quite different from the resting naive cells and from the switch memory. So this allows you really to validate your phenotypic separation of distinct subsets and then start really putting them together from a differentiation standpoint. So this is our summary of all the transcriptional data as to what we believe that the DN2 B cells represent. We think that they are effector B cells on their way to becoming antibody-secreting cells through an extrafollicular pathway. They are characterized by extinguishing the transcription factors that maintain the naive phenotype, so they are allowed to differentiate. They are missing the critical transcription factors of central memory or of the germinal center program, and they are driven by TBET and SEP2, which are co-regulated, as well as AIRR4, and that creates a plasma cell program for these cells. We can see that. I'm not showing you the in vitro data, but we can stimulate them in vitro. They are highly responsive to TLR7, as well as IL-21 and interferon lambda, and they differentiate into antibody-secreting cells. But this is to show you actually the in vivo analysis by repertoire sequencing and tracking. So again, these are two illustrative patients with massive expansions of plasma cells in the peripheral blood, massive expansions of the double negative cells that are actually the DN2s, as you can see here. So when we purify those cells from these patients and do the next-gen sequencing, and they match the repertoire, and we've done it both by bulk sequencing of the sorted populations as well as with George's approach of high-throughput emulsion PCR for single cells. You can see that depending on the assay and depending on the patient, the actual relationship varies, but it's very, very significant in this patient, for instance, with a few thousand single cells. Close to 25% of all the DN2s are related clonally to antibody-secreting cells. Some of that is illustrated here by the two methods and the different populations. You see that the DN1s in this particular case are not that related. This is just a few more samples, although it's heavy on the switch memory, but you can see that in multiple patients, actually, there is a major relationship with the antibody-secreting cells, as Chris had shown before. In the activated naive cells, the DN2s is also quite substantial, and then the switch memory, some patients more than others, so we need to match these compartments in the same number of patients. But it clearly contributes a lot to the circulating antibody-secreting cells in these active lupus patients. So I think that at this point, just to summarize all that part of the talk, we place the DN2 cells or through all the criteria that I just mentioned, here in their differentiation way from activated naive cells to short-lived plasmablasts through an extrafollicular pathway. There's another number of reasons for that I'm happy to discuss with you, including the absence of ETS1, which in the mouse at least is extremely important to block plasma cell differentiation. And when you actually knock out ETS1, which incidentally is genetic susceptibility loci for SLE in humans as well, these mice develop autoimmune disease in the absence of ETS1 and accumulate B cells and plasma cells in an extrafollicular location. On the contrary, we think that DN1s really are precursors of memory cells, and again, that's supported by all the analysis that I showed you. And we think that this is actually informative because one of the features I didn't show you, but is very prominent, is that these activated naive cells and DN2s express quite significant levels of SLAM F7, which is highly expressed in plasma cells. And in fact, elotuzumab, which targets SLAMF7, is being approved for the treatment of refractory multiple myeloma. And so we think that with this antibody, we may be able to eliminate all these components of the B cell compartment, as well as long-lived plasma cells. And we have now preliminary conversations with BMS to actually carry out this type of a study in SLE. Now, we use the same approach to try to find the origin of the B cells and plasma cells that are infiltrating, in other words, of the B cell repertoire that is present within the kidneys in patients of lupus nephritis. So we've done a number of biopsies and parallel analysis of the blood in these patients. I'm showing you here some of the first patients. And as you can see in this particular one, for instance, what we can see really is from the systemic circulation, a lot of connectivity of the top clones with the top clones within the kidney. This one in particular seems to be differentiating in different directions, so we are trying to analyze through clonal analysis what is the directionality of this clone and what the origin may be. Here is found in the double negatives, which is not, they are not best discriminated into DN1 and DN2s by this approach, so we are trying to understand which one is the origin of the clone initially. But clearly, the top clone in the double negative population in the periphery matches very well the top clone within the kidney, and also is connected with the antibody secreting cells. And this is just a summary of similar results in 5 or 6 biopsies, again, showing that as expected, the switch memory is providing a significant number of antibody secreting cells, or at least of the clones that are within the kidney, and the DN2s at the activated naive cells are also from the blood, all of them. So I think that there is a major systemic component rather than local production of these clones. Now, in general, one of the things that we have been quite interested in looking at lupus repertoires, and again, Chris published that 2 or 3 years ago, that's perhaps best summarized here, is that if you look at memory cells, for instance, or plasma cells, in lupus and normal controls, these are healthy controls immunized either with flu or with tetanus. What you can see, what this is showing here actually is the frequency of cells or clonotype within those patients in these populations that actually were scarcely mutated. So not much more than what you might expect by errors in the sequencing. And what you can see even within the IgG compartment is that in some patients up to 40 to 50% of all the IgG memory or even IgG plasma cells are really not much mutated at all. And again, to us, this suggested that there is a constant input probably of new reactions coming into the IgG memory and plasma cell compartment rather than just a re-stimulation of pre-established memory with very high levels of mutation. And this is now shown in here in very similar results, now with a much larger number of subjects. And if you concentrate here in the switch memory, again, you see something similar relative to the healthy controls, and then for the plasmablasts and plasma cells. This is another type of analysis that is similar to what Steve has described, looking at the length of the trunk for the memory B cell clones as a measurement of how recent or how close to a new memory cell before accumulates a lot of mutation over time may be. And as you can see here, the length of that trunk is much shorter in SLE memory cells than in healthy controls memory cells. So we take this to mean that, again, there is a lot of renewal or at least addition to the memory compartment in SLE that is starting to mutate rather than feeding from a pre-established memory and just continue to mutate that compartment. That doesn't mean that doesn't happen, but it really shows a different dynamics there. And this just to give you a sense, there is a table later to complement this because it was also brought up yesterday, and I think it's a very important point, that certainly within this community, there is a lot of resources as people start accumulating mutations or sequences, I should say, that it'll be very productive to share. And I'm just giving you a sense of what we are accumulating. These are studies of blood and bone marrow, both in SLE. We must be close to 30 bone marrows now in SLE, and then a similar number in healthy controls. And we have actually cells and sequences from the different sorted compartments. And this just to give you, this just says in here, the relative frequency here is not about sequence of mutation. This is just the distribution of those B cell populations in the marrow and in the blood of lupus in red versus healthy controls in blue. And this is a summary of the actual sequencing that we have for multiple samples over time. And you can see here the marrow, 20 and 23, kidneys that we continue to accumulate, and then the urine from the same patients, peripheral blood, mostly in lupus, but in a number of other situations as well. And these are all the samples here of sorted populations for which we already have repertoire sequencing that Chris has been doing over time. So there's a richness here of data that it could be helpful to other people as well, and that we would be happy to collaborate if people have questions for which this could be helpful. So the summary of this part is that the integrated phenotypic, transcriptional, epigenetic, and functional analysis define distinct B cell populations and their differentiation pathways. When you add the B cell repertoire analysis, then it helps to define their participation of the different populations in systemic and local autoimmune responses. We think that we can define through this approach the extrafollicular pathway of human naive B cells and how they differentiate into antibody-secreting cells through CD11C, high, TBET high, DN2 B cells. In contrast, we think that DN1s represent newly generated precursors of memory cells. We think that this pathway is highly prominent in active SLE, and I told you about the molecular program of the cells and what they respond to. We have nice data, I think, as to, in part, why they are hyper-responsive through negative regulators of TLR and NF-kappa B, but again, that's not the topic for today. So what I like to do in the second part of the talk is just to show you the studies that we are doing with the epigenetics of B cells in SLE. This is a combined effort with the Autoimmunity Centers of Excellence at Emory in collaboration with Jerry Boss. And this is an older slide, but we have done quite a few libraries for RNA sequencing, methylation through reduced representation bisulfite sequencing, and chromatin accessibility through ATAC6. So it's a massive amount of data that have been analyzed already independently and now is going through phase 2 analysis, integrating the different data sets. And I'll just show you a couple of illustrations of what we are seeing. So you can see here by methylation that all the different populations that we analyze seem to just lose methylation according to their differentiation stage, with the DN2s and switch memory being more differentiated and with less methylation. When you start looking at either ATAC6 or RNA sequencing, the populations start to separate in different principal components, and they become different from lupus to healthy control. And just as an illustration, when you look here at ATAC6, and mostly to the distal enhancers, which are more informative in terms of B cell or cell fate altogether, you can see how the DN2s and the activated naive cells become very close to each other in SLE, but not so in healthy controls, and also closer to the plasmablasts. So that starts allowing you to understand genetic modules that actually either close down or open up as the cells differentiate in either healthy controls or SLE, and there are distinct modules actually in SLE that we are now trying to understand better. So some of the transcription factors that we see that are most accessible in SLE relative to healthy controls are shown in here. They are very interesting because they are heavily involved in B cell differentiation, like CTCF, as you can see here at the top, but also the IRFs and AP1 factors that are critical for plasma cell differentiation. And I'll show you our model there. But just to make the point here very quickly, we published this last year that in fact, when you look at even resting naive cells before they start activating and differentiating, they are extremely different epigenetically between lupus here and healthy controls here. And once again, when you look at the top transcription factors that are open in those resting naive cells in SLE, they are also critically important for the differentiation of B cells into plasma cells. That's captured here in this scheme. What we think that is happening is that the lupus B cells have a lower activation threshold to go into the extrafollicular response through higher expression of IRF4, together with AP1 and IRF complexes, the ICES, and in the absence, I think that this is very important, of ETS1, which make this ETS-independent genomic targeting of IRF4, and then preferential association with the interferon-stimulated response elements. And that, as Harinder Singh has shown very nicely, actually sends the B cells into this extrafollicular plasmablast differentiation. So we think that that's what we are seeing in SLE, and that takes us back to our immunomics approach to try to understand whether the antigens that are actually stimulating this type of differentiation are different than the ones that send the cells into the germinal center pathway. And so we apply these that I was showing you in the beginning to now try to understand the repertoire of these cells. And just to give you some examples, this is what we showed before with the activated naive cells. I showed you this scheme before. This also addresses a point that was brought up yesterday by Jim Crow, but this is a nice example of that. If you actually analyze, what this is showing in here actually is the clonality of the activated naive cells, okay? And here what it's showing with the bars is what fraction of them are connected to antibody-secreting cells the same day in the blood. What you can see is that when you actually analyze the same patient a few days apart, actually the results change dramatically. So if you were to analyze only here, you would miss a lot of the clonality and the connectivity. But if you look systematically over a week or so, several times, then you will find a very different repertoire, okay? And this is just showing when we take those cells that was part of the paper, make monoclonal antibodies out of them, many of these naive activated cells already have a lot of autoreactivity to anti-nuclear antigens. They are enriched for the BH434 antibodies that we know are very autoreactive in general and in SLE. And we can start making some structure correlations with the activity of the antibody. We took advantage here a few years ago actually of the non-intrinsic autoreactivity of BH434 that depends, as most of you know, on the presence of a hydrophobic patch in the framework 1. So it's really an atypical type of binding, but it creates an almost universal reactivity against carbohydrates expressed by the blood group antigens and also naive B cells in the B220 glycoform. And so on that basis, we define autoreactivity that is dependent on that patch. Again, the B cell binding, B220, and red blood cells, and also ANA nuclear antigens, mostly RNA. And as you can see here, when you take that monoclonal antibody and then mutate just the idiotype part of it, that hydrophobic patch, so that they lose it, you see that the nucleolar reactivity goes away. That's also true here. So that's a very nice structural functional correlation of these autoantibodies from the germline to mutated ones. And this is actually an interesting paper just last year that Chris Goodnow wrote on the basis of this system. And essentially what he showed is that when you have an immunization of normal subjects, you do get some BH434 clones that responds to the antigen, let's say flu or whatever you are using. But you need to protect against autoreactivity. And the way it seems to happen is by mutation of that patch that provides autoreactivity. And he showed that with just 2 or 3 monoclonal antibodies in this really nice paper. So we had also shown that before, as we find these major BH434 clones here in red in some patients after flu vaccination subjects, actually normal subjects, or tetanus vaccines, right? And so essentially he's right. I mean, those clones have lost by mutation this patch, and essentially they are not autoreactive anymore. So we've accumulated all the sequences that we have and start analyzing that. And as you can see, perhaps more prominently here, when you look at the switch memory plasmablasts and plasma cells in healthy controls after vaccination, this is the frequency of cells, BH434 sequences, that have actually lost the patch or that retain it. So you see that is much, much lower than in SLE, where that autoreactivity seems to be selected. So these are very useful system in which you can ask really functional selection questions through the deep sequencing. And this is sort of what we had shown actually initially a number of years ago with more limited but still deep sequencing, showing that in fact in SLE, that's the red slice, actually is dominated by idiotype positive autoreactivity. So we can track that. And that is just also shown in here, how in a healthy control after vaccination, these are plasma cells. The majority of them are actually mutated in the hydrophobic patch, whereas in SLE, just random plasma cells are not. So they retain the idiotype. So that's a very nice information that you can get through sequencing. So to complete this part and the whole talk, we believe that the B cell receptor repertoire sequencing complements very nicely the phenotypic and molecular analysis and also benefits from it, because I think that outside that context, it's going to be much harder to establish rules as to clonal size or clonal evolution or the destination of those clones unless we can define the populations very well. And so I guess that's sort of what I'm trying to say here, but also the repertoire analysis, obviously, and mostly the single cell approach that provides the heavy and light chain combination so that now you can test the autoreactivity of the clones of interest, really can contribute to elucidate, which I think is a critical question in SLE, which is where are the actual antigens that trigger the cells initially versus the antigens that select them later on, which they don't have to be necessarily the same. And I don't think that is something that we understand. And then finally, as I showed before, and in part through this study with George, trying to understand also how the selection rules work to send the cells into the long leaf compartment versus more of a short leaf compartment as well. So just to finish with the acknowledgements, as I said before, Chris and Deepak have done the sequencing. Deepak is doing the single cell. Jen Holm as well with the next-gen sequencing. Scott has done the DN2 work. And a number of collaborators at Emory, at UT Austin, as I mentioned, George and Greg Ippolito, and the group at UAB, and in particular Alex and Chris that are here today, and who have been critical for our bioinformatics capabilities. And then, of course, Ramit Mer also with her IG3 analysis, and we have a very active collaboration with her that we think will be extremely productive as we continue to the next phase of repertoire analysis. So I'll stop there. Thank you very much, and I'll be happy to take questions.
Wow, that was a lot, Iñaki. I just wanted to ask you just another layer of complexity that you probably have thought about a lot. SLE has a T cell component to it that may be a driving force. Mm-hmm. And I was wondering how you would approach putting those two things, putting that together with what's happening at the B cell level, and what kind of dysregulation do you think could be playing out there, especially with these short-lived?
Yeah, so that's a critical question, right? And we are only addressing part of it. There's a lot of people working on this, although not necessarily putting together the B and the T cell compartment, right? But there is a number of, what we are trying to understand is what T cell interaction do these double negatives experience. And we are doing that in collaboration with a number of people, including Hide Ueno at NYU, Mount Sinai, and also with Marcus Clark within the Kidneys, right, in Chicago, just trying to see what is actually the connectivity and the synapses between these B cells in the tissue, for instance, and the different type of T cells. And there is a number of candidates. There is a number of at least 2 new populations of CXCR5 negative T follicular helper cells. It may be a misnomer, but anyway, that both Virginia Pasquale as well as Mike Brenner have this. Well, one is already published, the other one is not, and they are different from each other. But so there is a number of candidates, and I think that we need to approach this, for specific B cells in specific tissues, how they talk to each other, right? Now, for repertoire, I'm not even sure how to go there. But at least at the population level, I think that we should be able to understand what's driving the B cells from a T cell standpoint in lupus.
Thanks, great talk. You mentioned that when you would sample the same individual at multiple time points, even very, just days apart, you got different results on the clonality. Yeah. I'm wondering whether you think that's due to sampling issues, sequencing? Is it something with the disease dynamics that's changing over those times? Or
I would say all. Yeah, I'd say all of the above, right? I mean, I think that it makes sense to believe that if we are not exhausting the repertoire or the depth of sequencing, we are missing something. Whether that accounts for differences in clonality versus different clones at different times, that I don't know, but it has to be a factor, right? But the other thing I think is this is a very dynamic system, right? So in fact, I'm always surprised when we even find significant connectivity from the same blood sample, because you would assume that the B cell differentiating into the plasmablast, the naive cell, it takes several days at least to get there. So it doesn't make sense that it's going to be synchronized, right? And so now you have, half-lives and persistent and migration. So I think that the point is that the more you sample, the more realistic picture you get, right? And any single time point is always going to be, I think, the floor of what really is happening. So, I think that that's reality. Not very satisfactory, but I think that that's what it is.
Great talk, and thanks for the advertisement.
Thank you, George.
So is there a correlation, and this is somewhat peripheral, but actually related to what you talked about, is there a correlation between specific loci that are susceptibility loci and specific classes of autoreactive antibodies like C1q, BRAF, BLIMP, you name it? Are you more prone to get different kinds of autoreactive antibodies given a particular genotype?
So the easy answer is yes, and I think that that has been shown already for polymorphisms of IRF5 and STAT4 as well. And so I think that the easy answer is yes. Now, do we understand all the complexity of it? I mean, that was mostly for anti-DNA versus anti-RNP antibodies, right? There are many other autoantibodies in lupus, right? And then, not only that, there is, we always talk about lupus autoantibodies as if all the patients have the same complement of autoantibodies, but they don't, right? And so I think that we don't understand the granularity of all those relationships, but I think that there is already precedent to show that, yes, the genetic polymorphisms are influencing the type of autoantibodies that you get.
And along the same lines, is there any clinical, I don't know if any of the anti-complement antibodies, anti-C5, etc., are they being tested in lupus? And is there any clinical evidence that blocking complement, because that's a driver?
For therapy?
Pardon?
For therapy?
Therapy, right.
Well, some of them, I mean, eculizumab actually is being used for some refractory lupus, and it may have a role there at this point in refractory cases. But that's the extent of it, to my knowledge.
Has been used, okay.
Yeah, yeah.
When you match spectra, what is the percentage of spectra you are able to match? Is it as low as in recent studies, like below 5%, or you're able to match more spectra?
When you say spectra, you mean from the proteomics analysis?
Yes, mass spectra.
Well, I think that that's a question for George. Because, I mean, we do this in collaboration with him. I mean, before we did, actually, the 2 papers that I showed, we did with Gabriel Chang at the time at CST. So I don't know the answer to that. I mean, what fraction of the spectra of the proteomics information is matched to the cells? I mean, that's something I'm sure you know. What we believe we see, again, from our collaborators, is that what they consider to be, we did it with 94 in particular, the most abundant spectra or clonotypes in the serum seem to match the clonal expansions that we saw in the plasma cells. Thank you.
All right, for our final speaker in this session, happy to welcome Johannes Struck from the University Children's Hospital in Zurich.
Great, good, that's great, thank you. So first of all, I would like to thank the organizers for the opportunity to present some of our data here. And compared to the previous talk, this is very simple. So we have simple graphs and simple facts plots. And the idea, well, first I'll show you where we are in Zurich. If you ever make it to Europe, stop by. That's not a photoshopped photo. It's really like that, at least sometimes. Good. So I'm going to talk about repertoire analysis in patients with primary immunodeficiency. So these are, as you wish, human knockouts. The idea was to get something in addition to what we have, and we run into trouble with quite a bit of diagnostic testing, and the idea is really to have it as close as possible to the clinical setting, to take the sample, take it to a lab, do some testing, and get back to the patient. So I'm going to talk about some examples where we did repertoire sequencing of specific PID, so primary immunodeficiency patients. And I also give you a little bit of information of what we've done in terms of age-related changes. This has been sort of mentioned a few times yesterday and today, and I will make the point that this is crucial. This is very important. If you want to compare something that you think is abnormal, you have to know what is normal, and that is dependent on a few factors, and one of those is age. I'm also doing some work with patients post bone marrow transplant, those following up after transplantation, and see how the immune reconstitution works and how these cells basically come up and what comes first and so on. So I'm not going to talk about that. I'm focusing on the first 2 points here. So we're not the first ones who are doing repertoire sequencing in primary immunodeficiency, and we've put together some data on previous literature in that recent review, and I'll just guide you through. So at the top, can you see my mouse? Oh, there's a pointer. So these are basically all some well-defined diseases. This is mainly antibody deficiencies. These are combined or T-cell deficiencies, and each dot is one study, and the color indicates whether that's sort of a low resolution, being single cell, or spectrotyping, or these are next generation sequencing studies. And as you can see with next generation sequences, there are more studies that have come up, and there's one particular bigger study from Scott Boyd's group who's looked at CVID patients as very good work. So just what I'm going to cover today, just a little bit about primary immunodeficiency, that you know what I'm talking about, a little bit about what we do in routine testing. I'll tell you what we do in the repertoire analysis, and then I'll show you some findings. So this is a very basic cartoon of lymphocyte development. You all know that. It starts off in the bone marrow with some stem cells, and you get these cells in the periphery. We're going to focus on B cells, obviously, and this just illustrates the way, in a simple way, how a cell goes onto being developed into a mature, a naive cell, then in a germinal center, and you end up having those plasma cells secreting antibody, and those antibodies protect us, for example, from disease, or if they're dysfunctional, they may give us autoimmunity. And this cartoon also, I use this to give you some ideas what problems can arise in patients with primary immunodeficiencies. And we know for some of these diseases where the problem lies along the way, from here down to being a problem down here, and probably everyone knows the XLA, the X-linked agammaglobulinemia, where there's a problem with BTK, with the kinase, which is essential for lymphocyte, for B cell development. If you don't have that, you don't have any B cells, you don't have any antibodies, and that's clearly a problem. And along the way, you can have different problems that may affect the maturation, survival. Very interesting are the class switch recombination defects. Some of those are actually T-cell problems where the communication doesn't work, and therefore you don't have a germinal center, no switching, no mutation, and then you're missing that whole bit here. And I'm going to show an example of that one. So they all have in common that at the end, that what we see as a clinical phenotype, but also sometimes by the routine testing, that they don't have enough or dysfunctional antibodies. Dysfunctional meaning that they don't protect against what they should protect. And that sort of was the idea, that irrespective of what's sort of happening in here, you've got a problem down here, so why not look at the repertoire and use that as a diagnostic test or some indication for what the problem is, where it lies, and how severe it is. So I'll give you a little bit of an indication of the primary immunodeficiencies, and I'm not going to go through that list here. To be honest, that whole system is quite a bit of a mess, since all the genetic diagnosis, some of these diseases had to be shifted from one group into the other. It's a mixture of clinical phenotype, genetic, and something we don't know. But overall, there are patients who have problem with T cells or B cells. So these are the ones we're interested in when we look at the repertoire. Then there are others where there are more immune regulation problems, so autoimmunity, for example, which is often also antibody driven. And then there's something which is quite interesting in innate immune defects, where there's some B cell problems, which I'm not going to talk about. There's great studies about that. From my perspective as a clinician and as someone doing these repertoire analysis, I have a different categorization. I basically have healthy individuals. Then I have patients with an antibody problem. I know their genetic diagnosis. Then I have other patients that have something wrong with their antibody production, but I don't know what they have. This is the majority of the patients, to be honest. So CVID falls into that group. Then I have patients where we know there is a T-cell defect, but that affects the production of antibody, for example, these ones. And then there's ones which I call broader PIDs. These are often transcription factors where there's something globally wrong, and then it's quite difficult to find out what cell type has which effect on which other cell types. But often these patients also present with a B cell phenotype. And so I'm going to show you some data on that too. And finally, we've got those patients where we're not quite sure whether they actually have a problem or whether they had, it was bad luck. They, for example, ended up with a severe sepsis. And this is currently being an active matter of debate. And these are the most difficult ones, and that is where we want to get to in the end, and I can't tell you the answer to that today. So this is what these are the examples I'm going to show you some data on. So just to mention for the routine testing that you have some indications what we actually do when we see a patient in an immunology department. We can measure things that give us numbers, quantitative, and we can measure other things where we think it gives us some quality. And this is very easy, it's very cheap, you can do it very easily, and that gives you some indication whether you have good concentrations or low concentrations. Then you can measure the number of cells, and we've heard a great talk about all these different subsets you can measure. And then you can typically do some other things like measuring antibody against vaccines, against proteins or polysaccharides. And there are various ways which patients who can have a deficiency in one or the other, but it's often difficult to interpret. And then you can do some other T-cell assays that may help you in the diagnostic pathway. So again, the aims of the project was really to be close to the clinic, to have a workflow that is as easy as possible and rapid. We wanted to cover all isotopes because we know that is important from the serological parameters, all isotype subclasses. We wanted to have good coverage across these isotypes. And we already knew, and we have experienced that there are challenges with that type of setting, that you have to basically, if you have a 6-month-old child that comes once a year to your, well, no, once a year, or first time for investigation, you can't take 10 mils of blood for research. So you have to take what you get. And then an inherent feature of B cell defects is obviously that you often deal with low B cell numbers. So that is another challenge, and that might affect some of the analysis too. So very briefly, 2 slides on our method. So we basically take blood, isolate cells, do RNA extraction, our PCR, a library preparation, run it on the MiSeq 2 times 300 base pairs. We all do that in the lab. And just a quick note in our PCR that is stretching the amplicon quite a bit, but we wanted to have as much of the C region as we can, and it works, and we have a barcode, and so that gives us what we actually need for the analysis, and we can multiplex several samples here. So we had some issues initially, and I'll just show you what we did. We tried to improve the protocol in order to get better coverage across the different, so each box is an isotype. These are the first 4 runs that we did. This is number of filtered sequences that we got. And by doing some modifications, we actually can now get up to almost 100,000 sequences per isotype per individual, which is great. Although some of those will then be collapsed to unique sequences, but that is the depth we need in order to see the rare sequences there. In terms of analysis, this is, I think, pretty standard. We use some of the toolkits that are available. They're mapped to IgBlast. We use a genomic mapper for the isotype subclass identification because we believe it's good and it's very quick. And we have some other method of doing clonal clustering that we do in-house. So I've promised I'll show you some data on age effect on the repertoire. And the initial idea was to get some characteristics of the repertoire, have it in healthy controls and in patients with immunodeficiency, develop a score, and then basically you can use this score as a diagnostic test. One thing we realized, I mean, this is not entirely new, but to that extent, I think this is still quite interesting, is when you look at just the mean CDR3, so junction length, in here each box is a isotype, and this is along the x-axis from 0, so I think the youngest is a few months old, up to 40 years. Each dot is a healthy individual. You see there are differences between isotypes. So that's the first thing which you can probably notice, that if you would lump all those 3 Gs together, you would have something in the middle, but you would miss that distinction between the isotype subclasses. So that's the first one. Then there's an effect of age. It seems to be more pronounced in some than in others. And the third thing is that it doesn't go always in the same direction. So if you believe that and that, then even for IgA and G, it does go sometimes in different directions. So that's just to making the point. I don't know what that means, and I don't know why that is the way it is. But I just to highlight that we need to compare like with like when we compare some patient with a control. It's even more pronounced when you look at mutations. So this is the mean of a number of mutations. Obviously, D and M is low down here, and you've got the switched sequences up here. But there's quite a strong effect of age, and a little bit different to what has been published. We don't see, at least up to the age of 40, not really a plateau. Others have said that it sort of mutates up to 7, 8, 9, 10 years, and then there's a plateau. We need a bit more numbers here, but it doesn't look like that it's plateauing. So this is just to highlight the importance of age-matched controls, and all the data I'm going to show you later on will have age-matched control alongside that. Good. So I've got 3 cases I'm going to show you here as an example, and sort of what we can take out from there. So the first one is a patient with a CT40 ligand deficiency. It's a rare primary immunodeficiency where there's a non-functional CD40 ligand, so you don't have any germinal centers, there's no B T cell interaction. And so this patient is now an adult, but presented at 6 months of age with a typical opportunistic infection. And in the laboratory, you see so this is also called hyper-IgM syndrome. So you have a high concentration of IgM and low switch concentration, and the cells also bear rarely switched, isotype-switched cells. So this is a naive immunophenotype, and this is a patient who was diagnosed then later on. So again, this is what I said earlier. There's a problem here. You don't have anything of that one here. So I'm going to show you a few of those graphs in the next few slides. And just to guide you through, this bar means these are healthy controls. This one is the one patient. These are the different isotypes. And this is what I call a VDJ plot. So in the x-axis, there are the V genes, D genes, and the color indicates the J plot. And I like these plots because they give you a quick overview of the diversity, in a way, the frequencies, because these dots are also scaled to frequency. And you can nicely see that when you look at DNM repertoire of that patient, this looks exactly like the healthy controls, but here there's a lot of cells missing. What is probably more interesting that is not completely missing, but they're actually cells. And these, if you look at IgG1 here, these are well, I'll show you these are mutated, but they are diverse sequences in this patient who doesn't make any germinal center. So this is showing like a histogram of the mutation pattern in the different isotypes. Again, healthy controls, and here the patient. And what I told you just now is that actually, although you have very low numbers of cells, so the bar here goes to 3, you see mutated sequences, and here you also see mutated sequences in IgG, and probably overall it doesn't look too different to some of the healthy controls. Something that has been a debate and has been also these hyper IgM patients have been used to make a model of this particular subset of cells, and previously they were referred to as the unswitched memory or marginal zone or natural effector. Everyone calls them differently. These cells, whether they are, so these are cells that have a memory phenotype, but actually have IgM and IgD often on the surface. And these cells have been thought to be either developed separately from the germinal center reaction or actually as an alternative branch. And in our patient, we don't have a germinal center reaction. It is known that these cells actually occur in these hyper IgM patients. And I think we can see, so this is basically the same graph than before, but not showing the big peaks on the left side with the 0 and 1 mutations, basically zooming in. And I think we can see both evidence of mutated mutated IgM and D sequences, but also mutated sequences here, and supporting the idea that there is outside of the germinal center somatic hypermutation and class switching. So to sum this up on that patient, that is what I've just said, and it's interesting to see that we see in this patient a polyclonal rare population of switched cells. They are hypermutated, and they will also see quite a good number of mutated IgD and IgM sequences. And as I said, this is evidence that there's germinal center, class switching, somatic hypertension happening outside of the germinal center. Whether there is really going that way, this is showing that cartoon just before, whether there are separate mechanisms is unknown. Okay, so I'll come to the second case, which is a bit more, is one of those broad immunodeficiencies. It's a very interesting disease that affects several cell types. And just as a vignette, a 6-year-old boy who came to our clinic for recurrent fevers. He didn't really have any infections. Some of those patients have really severe infections, but he didn't. But he had persistent warts, and his mother had the same issue, and they were finally diagnosed with this disease, GATA2. So it's a haploinsufficiency, so heterozygous mutation in GATA2. And the classical immunophenotype is that you have normal immunoglobulin concentrations, normal T cells, but very low B cells, and NK cells, but still don't usually have bacterial infections. The other thing which is interesting that they lack dendritic cells. These more conventional dendritic cells, but also other dendritic cells, are very low in these patients. And one of the ideas was that this has to do with probably a altered repertoire, that they can't really present properly because they are lacking the cells to do so. But nobody has really done this work looking at repertoire in these patients. And just one quick slide on GATA2. There's quite there's lots of information here, but it's very important in the stem cell, as in a renewal, self-renewal of stem cell in the bone marrow. And it's very important for a number of different differentiation of different cells. There's some redundancy in some pathways with GATA1. That's why people believe you don't have any problems with platelets and erythrocytes, because this is sort of covered by GATA1. But there's an issue with monocyte development, dendritic cells, and NK and B cell development. And this is the fax block of one of our patients here, and it's typically referred to as a prematurely aged immunophenotype. So you lack the naive compartment. So this is too low for healthy individuals. You see a lot of these double positive cells, and you see a lot of switched cells. So that's why we thought that is interesting to look at. And again, a similar plot. These are actually now 2 children, the patients here, and these are 3 age-matched controls. Again, a similar graph, and you may or may not see that in contrast to the previous graph I've shown you, that there are definitely good number of switched sequences there, probably even more pronounced relative to the healthy controls. But there are some, if you want so, holes in here. You still see subclasses where there are very few sequences, and this is actually quite an advanced disease in this patient who's already been transplanted for my MDS and sort of refractory, well, basically low cell numbers, cytopenias across. So this patient has a very severe phenotype, and there's just very few cells, but of the very few, you have good number of the switched sequences. Again, looking at the mutation pattern in these patients, probably again to highlight that healthy control is not healthy control. So you see some things that are quite difficult to understand why that looks completely different in that healthy control. But that just highlights again the variability in biological samples, I guess. But you see again a good number of mutated sequences in this patient group, both in IgM, D, and GNA. So to sum this up for what we've done so far on this kind of patients, we show that overall there seems to be a quantitative problem. We have a predominance of antigen experience or mutated sequences, and they are polyclonal and display a rather normal pattern of mutations. This is consistent with what we see in what we know and what we see in the fax plot, and this is probably the reason why these patients get worse and worse clinically with age. They probably just don't have the capacity to respond to new antigens. Good, so the third case and final case, I'll show you again something which I would refer to as a broad immunodeficiency, which is a Job syndrome or STAT3 loss of function or hyper IgE syndrome. So again, this is a patient who's now an adult who had some issues when he was young, liver abscess, respiratory tract infections. He also showed typical signs of hyper IgE syndrome with the skeletal abnormalities. And this patient actually has high IgE. Well, that's how the name was given. But he also displayed low IgG2 concentrations in blood, and he's doing, I would say, okay on replacement therapy. And his fact plot looks like that, again, dominated by naive B cells. And very briefly, STAT3 is a very important transcription factor that is important for all kinds of immune responses. The loss of function is actually not a real loss of function, because loss of function is probably not, you won't survive, but it's an almost loss of function mutation and has lots of issues with infections, but also other problems which I've sort of touched upon. There's a diagnostic hallmark that is used in clinical testing, low Th17 T cells. And the B cell phenotype in these patients is probably not exactly clear, but it's believed to be associated with a T follicular helper cell differentiation and some problems with naive B cell problems. And you sometimes see in some patients problems with antibody production. So again, similar graph here. You see, actually, again, these are other healthy controls. You see a good naive compartment. This is actually the patient with the IgG2 subclass deficiency, and you probably see that this patient has lower number of sequences in these compartments compared to the other patient who doesn't show this immunophenotype, but otherwise that doesn't look too bad. On the mutation side of things, again, similar in this one patient, you probably see a lower pattern of mutations along with this IgG subclass deficiency. And again, zooming in, there's something interesting that pops up, and I don't know what that means. We have to investigate that, but this patient has a expansion of mutated IgD sequences, which you don't see in any of the other healthy controls or other patients, and not in any of the other isotypes. So there's, I think, a level of detail with the repertoire allows us to look at some of these features which we didn't expect, which we now see, and we need to investigate further. Again, summing this up, there are quite variability in the clinical phenotype. We also see that on the immunophenotype when we measure things. We also see that in the repertoire. But we can probably say that at least for that one patient, the IgG2 deficiency may be linked also to the level of how well they can mutate. It's not completely abrogated, but you can see that it's lower. And yes, that's what I've mentioned. We don't quite know what that means. And we'll investigate further. Okay, so finally, that was a great introduction to what we're looking at as well, is the 434 V gene usage, which you've just heard in the previous talk, which is associated with self-reactive antibodies. And we just asked very crudely, are there any differences in the usage of that gene segment across the different immunodeficient phenotypes? And we can do that very easily. And we can do that across isotypes. And this is what several healthy controls look like when you just look at the proportion of sequences that are using V434. And that is something that has been described before. You have it more commonly in the naive compartment. It actually goes down when you look at the mutated, the switched sequences. And this is thought to be, as we heard before, that is basically outnumbered. It's either mutated away from self, or it's just not stimulated in the way that it's outnumbered by other cells. When we look now at patient groups where we know autoimmunity is at least for some patients, a dominant feature, and just very easily look at the proportion of sequences that contain this V segment, we can see that at least some of those patients, CVID patients, this is again a disease that is CVID-like, I would call that, where also autoimmunity is quite a big issue. You see that these have much higher proportion of this V gene segment usage than the controls. And this is true for both the naive compartments, but also for the switched sequences. And if you do that as a control for other diseases where we don't usually see autoimmunity, you see that is more similar to health. So I'm not saying that all of these sequences are autoimmune sequences, but it's sort of trying to find a signature that can help us in a diagnostic pathway. Okay, I've said that. So, and finally, we have not just now, but realized that, when we think about these patients, that obviously we're dealing with strings of characters, and we don't know even though they may look normal on the mutation level, and also we don't know whether these are actually sequences that have a good and a functional product. And we have some collaboration with a group in Oxford, a structural biology group, that do model 3D structures, and we've recently put that into a review, and the idea is to find some measures for the more structural space and use it on these patients, trying to get just additional information that might help us to divide patients' controls and severity. And just as an example, that might be a very extreme example, but just to highlight something we've also put in the paper, these are two pairs of sequences. The first sequence pair has quite different annotations here, has a different CDR3 region, very little sequence identity, but actually has a very nicely overlaying loop, which looks structurally very similar to each other, whereas the other sequence that the pair we've chosen, identical annotations here, only 2 amino acids different in the CDR3 region, but very different loop, and therefore probably very different functionally, and therefore used. So this is just something that we're working on, that we've trying to do on large data sets, trying to find methods to pull out this structural information or to get some grip on it, and trying to use it and compare in the different individuals. And so with that, I want to sum up, and I hope I have told you the significance of at least age. There were a few other things that were on the list we've just had, but age is definitely something I have enough evidence to show that you need to take that into account. I have shown you that different immunodeficiencies actually show different patterns, and we're trying to find these patterns across now multiple individuals, bearing in mind that these are rare diseases, so we need to get those for for other groups. That there are differences between problems in the naive compartment, antigen experience. We believe that the isotype subclass annotation is so to the knowledge which sequences belong to which subclass is very important, at least for our understanding, and that we hope that we can add eventually some additional information and insight to the routine analysis and linking that to function. And yes, I've mentioned that already. And with that, I would like to thank all the people who were involved, and particularly Marie Greshy, who's not here today, is a PhD student working on that, Jake also, and all the clinicians and other groups that were involved, and the funders. Thank you.
Really nice talk. I was wondering, do you look at patients that are both those that have total knockout of protein expression, or also those that are hypomorphs that have expressed a dysfunctional allele of, for example, CD40 ligands? Since in some cases you have SHM going on, so could there be that you have a sort of a partially functional allele?
Yeah. Yeah, there are a number of diseases where that plays a role. I mean, probably the most easy one would probably be RAG deficient patient, where there is a quite range from almost no problem to very severe. The main problem is really accessibility, and these are very rare diseases. We have some patients where we at least have different clinical phenotypes, and some of the issues around the B cell compartment is either very severe or very minor. To link that directly to protein expression is sometimes possible. We don't have these patients at the moment, but that is something that is probably very interesting to see whether a direction is actually, you get half the way and full the way to understand whether actually you're having some real path there. Yeah, but we don't have these patients at the moment. Yeah.
And then just a comment. You mentioned VH434. I just looked at the list Martin gave me this morning. We have two new alleles identified of that gene, so that might be of interest to look at.
Okay, yeah.
Have you had a chance to do even a preliminary look at bare lymphocyte syndrome, where class 2 is missing?
So say it again.
Have you had a chance to have even a preliminary look at bare lymphocyte syndrome, where class 2 is missing?
No, is the short answer. so some of these very severe phenotypes, they get transplanted very early. We have, in correlation with the stem cell transplant unit, we have some patients who have been transplanted, who have either be it haplo transplant or some mismatch where they have not full chimerism. And some of these patients, we can actually isolate both donor cells and recipient cells separately. And in these cases, we might be able to look at actually those severe phenotypes, just looking at the residual cells. But again, something extremely rare. Very rare. Yeah, we don't have that at the moment. Yeah.
Were you able to isolate or detect any IgE expressing cells in the blood of the GATA3 deficient patient?
Yeah, so I didn't show IgE, even though we did it. It's usually very rare. Yes, we did. I don't know off the top of my head how many and how, but it looked diverse. But there, it's usually you've got like 2 handfuls or something, so very rare.
Yes, I thought it would be a nice opportunity to look at IgE.
Yeah. So in some of these, again, the IgE issue with hyper IgE syndrome is interesting. You see, nobody really knows what these cells are and why they are hanging around and why they are so pronounced in this disease. But yeah, so there are a lot of things which I haven't shown, and we're investigating it.
Really interesting data. I just had a question related to the V434 gene usage. So autoimmunity in CVID is restricted to a subgroup of the patients. It's not generally seen in all of the patients. And you showed that the proportion of V434 gene usage was much higher in the patients. Is this also reflected in absolute levels of the antibodies, or is it so that you actually have less reduced antibody levels for the other isotypes rather than that you have, so the proportion is just because the other antibodies are disappearing because they have generally low levels of antibodies, right?
Okay, I get what you mean. I don't know is the answer. It's probably difficult with that kind of data to actually answer that question 100%. You would have to normalize by cells and yeah, well, that's something we could definitely do. It would still, even if it's just proportional, it would still indicate that there's a sort of an intrinsic issue with getting rid of those sequences. And I'm not saying that these are autoimmune sequences. I'm just saying this is something that we can use as a marker.
So from the GLDB perspective, so germline alleles, your data shows that so even for the CD40L deficient patient, you still do see things that seem to be hypermutations. Do you have any patients in your cohort where you know that they are really completely deficient for CD40L or even AID, perhaps, in an hyper IgM syndrome? Are there any studies that try to correlate in these patients somatic hypermutation and class switch to get some idea about remaining AID activity? So all with the aspects, are the hypermutations that you see or seem to see there, are those true SHMs, or is this mismatch versus the database?
So I mean, some of these sequences are heavily mutated. I mean, these are very distant, like 20 nucleotides difference from the proposed germline. There are some other studies who have more patients in both CD40 ligand and AID deficiency from the Rotterdam group. They have looked at some of these features. I'm not entirely sure if these papers would exactly answer your question, but that was one of the focus of their research in these patients. I think they have like a handful of each. patients. With a lack of expression is again, when you do expression and CD40 ligand expression, you see, well, you don't see any expression, but does that really mean it's zero? Is it something you just can't really detect? You know how those things work. You stimulate, you get a FACS plot, and you say zero. Is that really zero, or is that 1%? It's hard to know, and whether that is actually expression means that it is functional, is really interacting, because there are others, mutations, which give you an expression, but actually don't give you any interaction. So I think this is difficult to answer.
All right, let's thank Anna and all the speakers in the session.
One reason we scheduled a long lunch here with the working groups need to get to work and lunch the posters, but also now that we've seen a lot of the old hands at producing and analyzing AIR-seq data, we had a little extra time here for people who are just getting into the field, might want to get some time with these people and talk to them about, you know, a lot of different issues. So we've kind of built some extra time into there, and hopefully the people will be open to that. So, but then at 1:30, people interested in the IP issues, we're going to have an informal discussion and start at 2 with the formal agenda. Okay.